So let us start the third part of this unit, where first we are going to discuss one molecule, that is metaclopramide. Metaclopramide, from this little part of the structure of the molecule can be understood. That is, this molecule has one amide. Metaclopramide, this molecule contains one amide, and at the meta position of this amide functional group there is one chlorine, and of course there are other groups are there in the molecule. Now this is antagonist at 5 a hydroxytryptamine receptor, means the serotonin receptor. Now let us consider the chemical synthesis of metaclopramide. Now this synthesis of metaclopramide starts with the orthotolidine. Now when orthotolidine is subjected to the nitration, then nitro group comes into the benzene ring, and where nitro group will come, it depends on the directive influence of NH2 group and the CH3 group. Now CH3 group is ortho para orienting, but and the NH2 group gets protonated, and then protonated NH2 group becomes meta orienting. So because of that reason we get the nitro group para with respect to CH3 but meta with respect to NH2. Now when it is subjected to diazotization, then and followed by heating with water, then diazotization means NH2 becomes N2 plus, and when water comes under heating condition, then N2 goes and OH comes. So this is an aromatic example of aromatic nucleophilic substitution. Now when it is subjected to the alkylation by using dimethyl sulfate and alkali, then the phenolic OH becomes OMe, and when it is subjected to the K-menophore oxidation, then we know that methyl group gets oxidized to the way carboxylic acid, the way toluene undergoes oxidation and gets converted into benzoic acid. Now it is treated with the thionyl chloride, means carboxylic acid gets converted into COCl, and then one nucleophile we bring, means we have to prepare the amide bond. So and this has the structural similarity, this side chain has the structural similarity with the side chain present in metaclopramide. So this is CH2, one carbon is attached with the NH2, another carbon is attached with the NET2. So HCl goes and we get the amide linkage. Now when it is subjected to the catalytic hydrogenation, then nitro group gets converted into NH2 group. Now it is subjected to the acetylation. So acetylation means with acetic anhydride acetic acid, NH2 becomes acetylated, means NHCOCH3 it becomes, and when it is treated with the chlorine, then ring chlorination happens, and again where the chlorine will come, it depends on the directive influence of already three groups present. This amide group is meta orienting, so chlorine will come at the meta position. This OCH3 group is orthopara orienting, so it has come at the para position, and this is also NHCOCH3 is all amide, but here linkage is not with the C double bond O, but it is linkage is with NH, so it is electron donating group, so orthopara orienting group, so chlorine will come at this particular position. So the directive influence of these three groups, the new chlorine has come at this particular position. Now when it is subjected to the hydrolysis, deacetylation, deprotection happens, and NHCOCH3 gets converted into the NH2. So this is the total structure of the metaclopramide. So we can see that amide linkage is there, and with respect to this amide linkage at the meta position, there is one chlorine atom is there. Now this is used as now we will see the mode of action. So for which purpose doctor gives a metaclopramide? Metaclopramide is anti-emetic drug. emesis means what na? Vomiting. So antiemetic means to stop vomiting or the vomiting sensation depending on the patient's condition. Doctor gives metaclopramide. So it is used to stop or control nausea, vomiting, and it is used for the gastroparesis means it is a condition when the food moves very slowly from stomach to duodenum, and it is used in the GERD means the gastroesophageal reflux disease means these are all related to the our GI tract. So where stomach contents rise up to the esophagus and gives complication. So usually from the stomach food it is food goes should go to the down, but from the stomach if it goes up and comes to one region which is called as the esophagus, then it leads to some medical complication so that is called as a GERD, gastroesophageal reflux disease. So in the reverse direction food starts moving from the stomach, so that condition also can be controlled by giving the metaclopramide. Let us go to the next. So with this we have completed the serotonin receptors and related antagonists. Now I will go to the new receptor, that is the histamine receptor. And already we know that in our body three types of histamine receptors are there: H1 receptor, H2 receptor, and the H3 receptor. But let us see where are those receptors. Means these receptors are activated by histamine, whether it is H1 or H2 or H3, all are activated by histamine. Now let us see about the H1, H2, H3. H1 receptors found in the smooth muscles of intestine, bronchi and blood. Means intestine means in the GI tract, bronchi means the air pipe which connects the nose to the lungs. In that one zone is bronchi, and it is also present in blood. H2 receptors found in the parietal cell, that is related to the anti-ulcer drug discovery of cimetidine and ranitidine. So this is the H2 receptors are present in the parietal cell of the stomach, and they are involved in the immunoregulatory systems. Now come to the last one, H3 receptors found in the presynaptic area, and it controls the histamine release and its synthesis. Now although there are three subtypes are there, but our syllabus mainly focuses H1 receptor and H2 receptor. Now when the histamine acting on the H1 receptor, means H1 receptor is present in the intestine, it is present in the bronchi, and it is also present in the blood. So what function happens, what is its role when histamine interacts with H1 receptor? So it stimulates the smooth muscles including bronchi, it relaxes the blood vessels, so BP falls down, and it increases the permeability, which leads to the escape of the plasma protein. So these are the three important. Fourth is the under extreme conditions, its excessive response leads to the allergy. So when the so what do we mean by the allergy? So as already I have discussed previously, that allergic condition is related to the histamine liberation. Means now allergy means what? Now, in response to foreign matters, means whether it is the dust particle, pollen, or the live like that our coronavirus, chemicals, microorganisms, food, antibodies are produced inside our body as a preventive measurement to fight against them. Now these antibodies interact with antigens by producing histamine. So that excessive response of histamine through H1 receptors lead to the allergic condition, and it sometimes may leads to the skin rash, redness, breathing trouble, asthma, edema, means swollen organ, etc. So by blocking histamine receptors, allergic condition can be controlled. So those medicines are called as the anti-allergic medicines, which are acting on H1 receptor as antagonists. Now H2 receptors, come down to this slide. H2 receptors are coupled with the adenylate cyclase, means it's a G protein coupled receptor, and they are stimulated due to the decrease in calcium 2 plus activated K plus conductance, means They are associated in HCl release in that already we know that is the H2 receptors present in the parietal cell of stomach is have active participation in the HCl liberation in the stomach. The H3 receptors, their blockage increases blood flow and also some sedative effect in the brain. Now let us consider the biosynthesis of histamine and all these things we have to consider in this slide. So let us see that histamine is widely distributed in our body and the highest concentration are found in the lung, stomach and skin. So lungs key H1 is and the stomach is H2. So the principal production takes place in the mast cell of connective tissues, and the histamine can be released from mast cell in the antigen antibody reaction and as a result of this in some person's body excessive histamine release due to the antigen antibody interaction may lead to allergy. Now it will be having two tautomeric forms. So nitrogen here that NH secondary amine is away from the means meta position it is, and in this it is ortho position with respect to the side chain. So it may what it is called the position can change, the position of hydrogen can change. So now let us consider the biosynthesis of histamine. As always, this neurotransmitter can be synthesized from amino acid histidine, and the histidine structure is like this, and when the decarboxylation happens, then what it is called the histidine gets converted into histamine. Now, when the histamine's work is over, then histamine will be inactivated by our body's own mechanism. So, histamine is at the center, and as already we have discussed, the such type of cases key two important things will happen: a combination of monoamine oxidase, and another one is the catechol orthomethyl transferase. So now in any sequence it may work, means first MAO then COMT, otherwise first COMT then MAO. We will get that histamine gets will get inactivated. Now see that this is when on this histamine monoamine oxidase reacts, then that primary amine becomes imine, and imine undergoes the hydrolysis. So we get the CH2 CHO, means imidazole acetaldehyde. One hydrogen of acetaldehyde gets replaced by imidazole, and then aldehyde dehydrogenase means it will undergo the oxidation and we will end up into the imidazole acetic acid. This is the inactive form of histamine. Now in the reverse direction, if it is the COMT. Now COMT means catechol orthomethyl transferase, but this is non-specific enzyme. So what happens now? It is not only catechol, on this imidazole ring also NH becomes NMe, and when the monoamine oxidase comes, then the side chain CH2 NH2 gets converted into CH2 CHO. On aldehyde dehydrogenase, it gets converted into the aldehyde, undergoes oxidation to give carboxylic acid. So we get the N-methyl imidazole acetic acid. So there are two inactive forms of histamine. One is the imidazole acetic acid, another one is the N-methyl imidazole acetic acid. Now we will move to the chemical synthesis of histamine, which starts from the imidazole ring itself. Now when it is treated with the benzyl chloride, that NH goes in the form of HCl and N-benzylation happens. Now when it is treated with the formaldehyde, I have already told you formaldehyde is hydroxymethylating agent, means CH2OH group will come at the meta position of this with respect to both the nitrogens. Now when it is subjected to the catalytic hydrogenation, then that it is not catalytic hydrogenation, it is hydrogenolysis. Deprotection happens, and then when it is treated with the nitric acid, here nitric acid is acting as oxidizing agent, so primary alcohol becomes aldehyde. Now it undergoes the condensation, and finally there is a condensation that active methylene group in between nitrogen and C double bond O undergoes the water elimination with the form the aldehyde, and these two parts become one molecule, and when it is treated with the final step key HI red phosphorus followed by decarboxylation, then the side chain becomes CH2 CH2 NH2, means in this way we prepare the histamine, that is the chemical synthesis. Now we will go to the H1 receptor antagonist, where the name of the molecule is a chlorophenamine. So from this it is clear, from this name at least partly we can make out the phenyl group is there, chlorine atom we can be able to make out, and the amino group is also there. So chlorophenamine or the chlorphenamine, phenamine, whatever it is, the structure is same to same, which we will go synthesize. So that the synthesis of this obviously it is a chemical synthesis. Chemical synthesis starts from the para disubstituted aromatic compound, where with respect to chlorine para position ki CH2CN group is there. Now when it undergoes the reaction with the sodamide, then sodamide is a base and benzylic position ki anion gets generated. Now, when this anion reacts with the 2-chloropyridine and the chlorine goes, nucleophilic substitution happens, because and the pyridine ring comes at the benzylic position of this particular compound, so we get this. So now again it is treated with the sodamide, and means this is again that benzylic position, the leftover hydrogen, one more hydrogen is there, that hydrogen goes when it undergoes the reaction with the sodamide, and then we bring what it is called the that nucleophile NMe2CH2CH2Cl. So HCl goes, and that particular benzylic position, that it what it is called, that nucleophilic substitution happens, and at the last step what happens when we do the sulfuric acid, means cyanide undergoes hydrolysis. Not only that, decarboxylation also happens at this particular center. So in this way we can be able to synthesize the chlorphenamine, means the benzene ring is there, chlorine is there, and the side chain ki amino group is also there. So now, as already we have discussed, that is the mode of action is what it is, the antagonist. H1 antagonist means definitely it will be acting as anti-allergic. So anti-allergic medicine is the chlorphenamine. So chlorphenamine binds to the H1 receptor and blocks what it is called the natural agonist. What is the natural agonist? Or the endogenous agonist neurotransmitter histamine at H1 receptor. Now the allergy condition, initial contact of allergen. Allergen means a foreign matter with the B cell. B cell is our immunity immune cell. It has very important role. B cell, it interacts with the allergen, and the message goes to the plasma cell, mast cell, memory cell. These are all the cells of our immune system, and the release of antibodies happen because they have to tackle this allergen. So antibody gets released, and this antigen antibody interaction is sometimes excessive release of histamine happens, which leads to the allergic condition. Now this condition can be tackled by giving chlorphenamine type of anti-allergic medicines. Now what is its mode of action? This is the cell membrane, this is the H1 receptor, and in this receptor binding site there is the drug molecule chlorphenamine interacting with the binding site. Now when the histamine, the endogenous agonist at this particular center, when it comes histamine, there is no scope for interaction, so biological response will not go, and the allergy will be under control. Now all these neurotransmitter related these things are over, receptors are over. Now we will go to the hormone receptors. Their introduction to estrogen receptors, means estrogen is the female hormone, and we have that in our syllabus, so we will discuss all these things. So let us see what happens. It's different than the receptors associated to the neurotransmitters, because it is hormone receptor. Now when estrogen receptor is a protein, obviously receptor is a protein present inside the cell. It is not present in cell membrane, it is present inside intracellular part. When estrogen circulates in, estrogen is a hormone, so this hormone originates from one place, it mixes with the blood and it travels in the other part of the body. So they interact with those cells which have the estrogen receptors. Now let us see this diagram. This is what it is called the I have drawn the blood vessel, and their hollow circles are there. That hollow circles are a symbolic representation of estrogen. So estrogen hormone is flowing through the blood, and estrogen has come inside the cell, and where the estrogen receptor is there. So now you see this is the cell inside where nucleus is there. It is not cell membrane, it is nucleus and inside. So in this cell, say, take it for granted, we have the estrogen receptor. Now this is and the cell with the intracellular estrogen receptor. It is present inside the cell and it can interact with estrogen. Now this cell, you see here the same picture is there, but here estrogen receptor is not there. So this type of cell cannot interact with the estrogen. So all the cells will not have estrogen receptor. The cells which have the estrogen receptors, they can interact with the estrogen, number one, and number two is it is not in the cell membrane, it is present inside the cell. Now estrogen, next come to this, estrogen binds with the estrogen receptor and forms a complex. That complex interacts with the specific site of DNA, and the RNA is produced which guides new protein synthesis. So let us see the whatever is written in these previous 4 lines. The pictorial representation of this is this, that estrogen is coming inside the cell which have the estrogen receptor. It is forming one complex, and that complex is binding with the DNA, and it gives the signal, specific signal to the new protein synthesis to RNA. Now take it for granted, this is happening in the healthy cell. Now take it for granted that for discussion purpose we are taking the cancer cell. Now in cancer cell, tamoxifen interacts with the estrogen. Tamoxifen is a medicine which interacts with the estrogen receptor and stops estrogen to interact with the estrogen receptor. So it puts brake on the cell. So what happens? A new protein synthesis will be on halt, because estrogen will not be able to interact with the estrogen receptor, because the one drug molecule by name tamoxifen is interacting with this, so the message will not go to it. So this is a complex formation will not happen with the DNA, and DNA will not be able to send the message to the RNA for the new protein synthesis, so the cancer cell will not grow. So now what we have discussed, so this is what it is called, the tamoxifen is interacting with the estrogen receptor, and estrogen, there is no scope for estrogen to come. So this is the structure of estrogen. It is a hormone A, B, C, D, tetracyclic ring is there, and the D ring is what it is called, the cyclopentan ring. Now this is the tamoxifen estrogen receptor complex binds to the DNA, and it does not give the signal to the new protein synthesis. So the cell proliferation does not happen, and the cancer becomes under control, and it is a very good medicine for breast cancer. Tamoxifen is a very good medicine. Doctors very frequently prescribe for the breast cancer as one of the life-saving medicine. Thank you.